So this lecture is about the cell cycle, so think of it kind of like the life cycle of a cell. So the cell cycle is defined as the life of a cell from its formation until it divides. So again, kind of like its entire life from birth until, not necessarily death, but it's going to be division until one cell splits into two. Cell division is important in reproduction, so making more cells, in growth, and tissue repair. So if you think about, from the time you're a baby to when you're an adult, the reason why your bones get longer and your skin gets bigger and your muscles get longer is because they're replicating. They're going through cell division. So the more that you make, the more that cells divide, actually kind of the longer your bones get, for example. So reproduction is really important, splitting of the cells, making more of them, and then growing and development of the cells as well. So one big important piece of this cell division in the cell cycle is going to be DNA. So this is kind of our first foray into DNA. A genome is all of the cell's genetic info, so that includes its DNA. So the genome is the entirety of the DNA that a cell contains. Remember, in prokaryotes, DNA is simpler, so it's usually a single circular chromosome. Remember, the DNA in a prokaryote usually isn't in the nuclear envelope, so it's not as protected as in a eukaryote. In a eukaryote, you're going to get more than one linear chromosome, so it's going to be more of a line. The number of chromosomes depends on the species. So humans, for example, have 46. Mice have 40, a fruit fly only has 8, so it really depends on the species that tells you the number of chromosomes in each. And again, all that chromosome is contained in the cell nucleus, in the nuclear envelope of a eukaryotic cell. The most important part of cell division is going to be copying that DNA, because that DNA is necessary for all the cell functions. So taking that into account, before a cell can split, it has to split and copy all of its chromosomes. So before cell division can occur, the very first step is going to be to duplicate or copy all of those chromosomes. So definitions are going to become really important here. So what you're looking at here is a duplicated chromosome. So duplicated means 2 copies. So this part here is going to be called a chromatid. And usually a chromosome is made up of 2 chromatids connected by what we call a centromere. So a centromere kind of connects the 2 chromatids together. So what this is going to kind of look like is there is a chromatid, there is the other chromatid, okay, and that forms the characteristic X shape that you're used to with the chromosome, and you have the little centromere that kind of connects them in the middle. So these definitions are going to be important, that you understand the difference between a chromatid and a chromosome, that you understand the function of a centromere. So you see here, you start off with a single, right, chromosome, single chromatid, then it duplicates, so now we have 2 copies, and then what's going to happen is when the cell divides, each new cell gets a new copy of the DNA. So when we talk about your body cell, there's 2 main types of cells. This is for you in most vertebrates and mammals, animals. Somatic cells are what we call just general body cells. So that's your skin cells, your heart tissue, your muscle cells. All those are considered somatic cells. And then the other type is a gamete, which is going to be strictly your sex cells. So there's more somatic cells in your body than there are gametes. But gametes are really important because that's how you're doing sexual reproduction. So somatic cells are what we call diploid. Again, new vocab word. Diploid is 2N, which means there's 2 of each type of chromosome. So in a somatic cell, you have 2 copies of each chromosome. Somatic cells divide by a process called mitosis, and humans have a 2N or a diploid number of 46. So what it is humans have 23 types of chromosomes, but 46 total because you have 2 of each type. And then gametes, again, are going to be the sperm or the egg. So in men, it's called sperm. In women, it's called eggs. These are what we call haploid. So you're going to think diploid di 2, haploid half 1. And the haploid symbol is just an N. So remember, humans have usually 2 copies of each one to get 46. And a gamete, you're only going to have 1 copy of each one. You're only going to have 23. And what happens is a 23 haploid joins another 23 haploid to make a diploid cell, which then is what we would call the zygote. And then gametes divide by a process called meiosis, which we're going to talk about each one. So this is a very common picture, a very common graph that AP likes to use. I've seen them put this on a test, so you need to be really familiar with this graph. This is the cycle of a cell. Notice it's a circle, so there really isn't strictly a start or an end, but usually we start and end with mitosis. So you can either call mitosis the end or mitosis the start. And it's a circle, so you're kind of right either way. You go into G1, and then S, and then G2. And then G1, S, and G2 all make up interphase. And then the very small section here is the process of mitosis, so cell splitting. And then you have a small little wedge of cytokinesis, which is the splitting of the cytoplasm. So as you can see, most of the time, the cell is in what we call interphase. That's kind of when it's just living its life. It's digesting material. It's kind of making energy from food. It's kind of doing its thing. It's going to spend a very small amount of time in the mitosis phase, actually copying and splitting. The G's, we kind of think of them as grow, G for grow. So this is where the cell is growing, and then it's going to grow here too at the end. And then the S phase is going to be DNA synthesis. Remember, to synthesize means to make. So DNA synthesis, this is where the DNA is getting copied in S phase. So the mitotic phase alternates with what we call interphase. So remember, interphase is like the in-between phase. That's where the cell's just going to be kind of growing and living its normal life cycle. And then the mitotic phase is the splitting phase. So generally the order is the cell's going to be in G1, kind of grow 1. It's going to go to S, where the DNA is split. It's going to go to G2, where it's preparing for mitosis and kind of growing and setting up the organelles it needs for that. Mitosis is the actual splitting of the DNA. And then cytokinesis is splitting of the cytoplasm or the rest of the cell. And then once cytokinesis is done, you should kind of have an arrow that goes back to G1. So then it kind of goes back to the G1 phase. Remember, we did talk about one of the signals for a cell to split is its size. So we talked about that surface area to volume ratio. So some of that's going to be brought in here. So none of the stuff we covered is off the table. It's all going to be recycled. So again, interphase is about 90% of the cell cycle. It's most of the time that's what a cell is in. In the G1 phase, the cell is going to grow and carry out normal functions. In S phase is, again, where the duplication of the chromosome, you're going to copy that DNA. And then in G2, you're preparing for cell division, so you're kind of growing, you're getting the organelles in place for that, etc. Then the M phase is where the mitosis, the nucleus divides, and cytokinesis is where the cytoplasm divides. Now, one of the things that AP loves to talk about, even more so than just your general knowledge of the cell cycle, is the idea of these checkpoints. So a checkpoint is exactly what it sounds like. It's a point in the cell cycle where you stop and you check to make sure everything is going okay, the processes of the cell cycle. So it's a control point where stop and go signals help to regulate the cell cycle. So this phrasing here is really important. Regulate the cell cycle, okay? They like to talk about how the cell cycle is regulated, and that is done by checkpoints. You can see here there's 3. So there's one at G2, there's one at M, and there's one at G1. So these checkpoint ideas are really important, so you need to pay attention here. So again, there's 3 major checkpoints, G1, G2, and M. So G1 is the most important one. This is going to control the cell size. It's controlled by growth factors in the environment. So there's going to be 2 signals that are going to be here. It's either going to be the go, and the go means the whole cell cycle is going to be completed, including the replication. So that's going to be kind of, you're going to go from G1 to S to G2, that whole cell cycle. But you can also do a stop. And if the cell hits a stop at G1 checkpoint, it doesn't die. What it does is it enters this what we call non-dividing state, or G0 phase. So it's kind of like a G sub-zero phase. What that means is it's just living its life forever. It's not going to go into preparing for cell division. It still performs all the functions of the cell. It's doing all the things that a cell does. It's just going to kind of be in that, living its life kind of setup. Nerve and muscle cells stay at G0 forever. So that's why, your nerve cells, if you damage your spinal cord, if you damage your nerves, they don't regenerate. They never, once they grow, once they form, that is all the nerve cells you get for your life. Liver cells, though, can go back and forth. So you can have your liver cells. Most of you right now, your liver cells are all in G0. They're just living their life. But if you had a cut in your liver, or if you lose part of your liver, that cells can switch from G0 back into the go signal and complete the cell cycle. The G2 checkpoint is going to be controlled by DNA replication. If you have an issue with the way the DNA was replicated, if it wasn't done correctly, if you have mutations, if you have, issues in the cell size, if all that's not ready to go for the splitting, it doesn't happen. So the G2 is going to kind of check right before the cell is ready to split, make sure the DNA was replicated correctly and all that stuff. If it detects an issue, then again, the process is stopped. And most likely what would happen is the cell would go through apoptosis. And then finally, the M spindle or N phase checkpoint, it's going to check the spindle fibers, the microtubules that pull the chromosomes apart at the kinetochores. Kinetochores is another word, one of those vocab words that we're going to use. That's the little middle point that, is pulled apart. It's going to make sure that the DNA was separated, so you're not having, 2 copies on one side and 0 copies on another. It's going to kind of do its best to make sure that all of that went properly. Again, if any of these go wrong, usually what happens is the cell is going to apoptose. It's going to go through apoptosis because whatever processes it was trying to do didn't go correctly. So this is just showing you a picture of either the G1 saying go, which is meaning the cell cycle is going to continue, or this stop where the cell is going to go into what we call the G0. It's kind of like just a living its life type of phase. This is showing the end spindle checkpoint. So you're going to find out one of the phases of mitosis is going to be all the chromosomes lining up in the middle, and then they get pulled apart by these little fibers here. They're kind of made of the same thing, the microtubules. And what's going to happen is the kinetochores is a little connection point here. So kind of like fishing, the centrosome is going to send out these filaments, these fibers, these microtubules. They're going to kind of hook or attached to the kinetochore, and then on either side, they're going to separate and spread apart. If this attachment to the kinetochore doesn't work out, or the chromosome isn't split, like you get 2 of the same chromatids on the same side, that's what's being checked for at this checkpoint here. Your body has internal regulatory molecules. The 2 big ones that we're going to talk about are MPF and cyclin. Cyclin should make sense. Cyclin, like cycle, right? Going through a cycle. Notice that both of them peak at the M phase. So you get like lower activity in both during G1S. G2, it starts to go up, and they both peak at M phase. Kinases are another type of regulatory molecule that you've heard of before. We've talked about kinases before. In particular, one that is called cyclin-dependent kinase, or CDK. So this is a specific kinase molecule. And these are protein enzymes that control the cell cycle. They're activated with the cyclin. So the kinase and the cyclin kind of work together. And then cyclins are proteins which attach to the kinases to activate them, and the levels are going to fluctuate within the cell. So basically, if you have high cyclin, you're going to have high kinase activity. They kind of go together that way. The other internal regulatory molecule is the MPF, which is the maturation promoting factor. It's a very specific cyclin CDK complex, which allows cells to pass G2 and go to M phase and go ahead and split. So is the maturity there? Does the cell have all the pieces it needs? Is the centromeres in place? Is the centrosomes in place? All of that. That's what the MPF is trying to help it do. Notice it's kind of like a pairing. You have the cyclin and the CDK. When they come together, it's called MPF, and then the process kind of goes through. The levels of all these regulatory molecules is controlling the cycle of the cell. So those 3 that we just talked about are what we call internal regulatory. There's also external regulatory. So external is things on the outside of the cell. Those 3 examples here would be anchorage dependent, density dependent inhibition, and density dependent inhibition. So anchorage dependence is like an anchor, like where the cell is. This has to do with why you're not going to grow liver cells in your ear, or you're not going to grow heart cells in your leg. the location of a cell kind of dictates where it's going to anchor and where it's going to grow. So you're only going to get liver cells next to other liver cells. The density-dependent inhibition is where, and this is true, if you had kind of a plate here and you put some cells, they will grow and divide until the entire surface is covered, and then they'll stop. So what happens is the neighboring cells kind of tell the other cells, hey, we're full, we don't need any more, let's stop growing, and they'll go into that G0 phase. So notice the whole surface will be covered, but then it'll stop dividing. If you took a piece, like if you scraped like a hole in the middle, they would start dividing again, the cells kind of close to the hole until the hole was covered up, and then they would stop. This is kind of what happens when you get a cut. So normally your skin cells are kind of in a G0 phase. Sometimes they do, replicate because of the apoptosis. But if you got a major cut in your skin, so you had a major opening, think of it like scraping the cells, all the cells in that surrounding area would immediately click from going from G0 into G1, G2, and they would replicate to cover, close the hole. Then once the hole was there, you had the scarring and the scab was formed, then they'd stop. They'd go back into G1, G0. So again, it's kind of like telling the cell when to turn on and off. One thing we've noticed about cancer cells, that's showing you the cancer cells here, is they're not paying attention to some of those regulatory factors. Sometimes they're not even anchorage dependent. That's why that's called metastatic. That means it has moved around. And then they're not paying attention to this density thing. So they know that there's other cells there and they continue to grow. So we kind of have figured out part of the reason the cancer, and one of the big contributing factors to the cancer, is this regulation. Some other external regulatory factors would be growth factor, which is a protein released by other cells to stimulate cell division. So again, if you got a cut, that is when the growth factor would be released. So it kind of tell your body, oh, we need to close this hole. You also have a lot of growth factor when you're young because your body's growing and you're going to need more cells to replicate in order to kind of grow your body. Density-dependent inhibition is when you're going to have the crowded cells stop dividing. The cell surface proteins kind of inhibit growth and tell the cells, no, we don't need to grow anymore. And then anchorage dependence is the idea that the cells must be attached to another cell or the extracellular matrix to divide. Again, that's why you're not getting liver cells in your neck or something like that. So when you do your project next, you're going to see more in depth about mitosis and meiosis. I just wanted to go over a couple of the differences. One, mitosis is for somatic cells. So it is when you're making an exact copy. You are literally duplicating the cell. Meiosis is only for gametes. So meiosis is only used to have the DNA. So if you trace, you'll see that you start with, 2 purple, 2 blue, and they split so the new cell has 1 purple, 1 blue, 1 purple, 1 blue. Mitosis is where you start with a diploid cell and you end with a diploid cell. In meiosis, you see here you're starting with 2 purple, 2 blue, and you end up with 1, 1, 1, and 1. So 4 cells with 1 haploid. So again, diploid ends in 2 diploid cells. Meiosis ends in 4 haploid cells. The processes are similar. Some say meiosis is mitosis twice, but you're going to go again into more detail on this with your project. There are 2 main types of reproduction, sexual and asexual. So asexual is when you're producing clones. You're getting an exact copy of the DNA. This is usually when you have a single parent and with little variation. So you're literally cloning the DNA, an exact same copy. The good thing about asexual reproduction is it's very fast and efficient. You don't have to mix the DNA or any of that stuff. Mitosis is actually asexual reproduction. You are making an exact copy of the DNA. And again, we usually use mitosis to make more liver cells or more, body cells. Other examples of asexual reproduction would be binary fission. That's how bacteria replicate. And then we have sexual reproduction. This is where you're exchanging materials, you're exchanging DNA. This is what meiosis does. So meiosis is going to produce gametes. Gametes, remember, have half the DNA, and then the 2 parents are going to come together and mix their DNA. This results in lots and lots of variation, lots of different traits. It is slower and consumes more energy, though, than asexual reproduction. And this is how humans create more humans, and also some trees as well do this. So this picture here is showing you a hydra. So here's the bud. Here's the asexual reproduction of the hydra. And then here we have redwoods, which they produce actually sexually. So we went over this a little bit in the beginning, but it's important to know these chromosome counts and these words. So again, a somatic body cell in a human is going to have 2N, diploid number of 46. That means you have 23 pairs of 2 to get 46 total. We say that each pair has a homologous chromosome, one from each parent, homo meaning the same. So you're going to have 23, if you want to think, traits, and each trait is made up of 2 homologous chromosomes. Autosomes, those are non-sex cells. There are 22 pairs of chromosomes that do not determine sex, and then you have 1 sex chromosome where you're going to get either the X or Y, whether you're male or female. Gametes, again, are what we call haploid. So human gametes have an N, a haploid number of 23. So they have 22 autosomes and 1 sex chromosome. So if you're going to be an egg gamete, you have 22 autosomes and 1 X. That's going to be the girl sex chromosome. And sperm have 22 autosomes and 1 X or a Y. So in order to be X, Y, and be a boy, you actually have to get that from your dad. This is just showing you the picture of the homologous duplicated chromosomes. The red ones here would be from mom, and the blue ones are from dad. A karyotype is an image, a picture of an organism's chromosomes. So this is a karyotype here. They do this for genetic DNA testing. The karyotype is going to have, again, 23 chromosomes for a human, and here are the homologs. So this is like, the first set of chromosomes, and here's the 2. So there's 23 sets, 46 total. The sex ones are found down here. So again, usually you have to make a karyotype. So this is what, if you would get a DNA sample from a person, it would look like this. And then what the geneticist will do, this is genetic science, would have to come in and sort these chromosomes. And once sorted, they look like this. So you would take all those pairs, match them, and you can see here again, you have 23 sets of 2. That's how you get 46. And the question here is, how do you tell if it's male or female? Well, again, the sex chromosomes are the last 2. So right here, you see that this person has 2 X's and 0 Y's, so they would be XX, which is female. And again, if we do the same thing, notice 23 pairs of 2 for 46 total. What is the sex of this? Notice they have an X and a Y, so this would be male. Karyotyping is how we tell and can identify genetic abnormalities even when a woman is pregnant. So what they'll do is they'll take what they call an amniocentesis, which is the fluid in the amniotic sac, and that fluid has the same DNA as the baby. And they karyotype the DNA, and they try to see if they could predict that the baby's going to have any abnormalities. So if you look closely right here, okay, instead of 2 pairs, 2 homologous pairs on chromosome 21, you have 3. So this is called trisomy of chromosome 21. You have 3 full chromosomes when you should have 2, and trisomy of 21 results in Down syndrome. So this would be someone who's going to be born with Down syndrome. In general, you can see that 21 has a small size in terms of the chromosome. That means it generally has less genes. It's responsible for less DNA. The bigger the chromosome, the more DNA is on it. So if you have an issue with one of these bigger ones, usually those are not survivable. That would result in a miscarriage for a woman. So if you have an issue like this, if you have trisomy of 1 through probably about 18, definitely 1 through 12, look how big those chromosomes are. If you have an extra set of those, that's going to be fatal to the fetus. It's just too much genetic issues. Those are things like developing the heart and the brain and essential organs. If you have some issues with the smaller, lower ones, usually those can be survivable, and you have some characteristics of people with have some of those genetic abnormalities. This is just a total random thing that happens. What actually happens is the chromosomes don't separate correctly in meiosis. So instead of only having 1 copy, one of the eggs or the sperm results in 2 copies, and that's how this can result. And we're actually going to talk way more about genetic abnormalities and DNA and karyotypes when we get to the genetics unit.